Okay, welcome to Scientific Method. We're going to do a little introduction. For some of you, this will be review. For others of you, it might be more new. But it's a great place to start because we use Scientific Method all semester long. So let's start with the basics. What is science? I'm not going to read all the boxes to you because I think you're more than capable of doing that. But what we look at here is what is science? What are we trying to do? And basically, we're trying to explain the natural world. We're trying to answer questions that we see within that world. We propose explanations for what we see or what we don't understand, and then we attempt to test that sort of explanation. So what we're really doing is we're trying to organize the natural world. We're trying to explain it. We're trying to use evidence to support it and to explain it. How do we do it? Well, we start with an observation or a question. It's that gee whiz, it's that how come, it's that I wonder if sort of thing. And what we do is we gather data. So we make observations and then we gather data about those observations. And two types of data. You can say data or data. Either is acceptable. Two types are quantitative and qualitative. So when we talk about quantitative data, we're talking about numbers. We're talking about things that we can count, things that we can measure, things that are very objective. If I hand a bag of marbles to one person and ask them to count them, they'll come up with a number. If I take that exact same bag of marbles and hand it to their best friend, they should come up with the same number. Qualitative data are descriptions. They involve characteristics, things that can't be quantified or counted. So in that bag of marbles, there are varying shades of blue. And I hand it to the first individual and they may tell me that there are so many dark, medium, and light colored blue marbles. But if I hand it to the next person, they may take that gradation and their dark blue may not be the same as the first person. So we're talking about descriptions as opposed to something that we can count. A big thing, a big item in this concept of scientific method is a hypothesis. And what is it? So basically, a hypothesis is an explanation. It's a proposed explanation. What makes it a hypothesis is that it has to be testable. We have to be able to test our explanation, okay? We sometimes call them an educated guess. That's the most common thing that my students tell me that they've heard a hypothesis called. And that's going to lead us to the whole point of this little video is scientific method. And what that is it's a series of steps that scientists use to test a hypothesis. For example, to solve a problem or to answer a question. There are 6 steps that we're going to look at in scientific method. We're going to start with an observation or asking a question. We're going to form a hypothesis or a possible explanation. We're going to design a controlled experiment, and we'll talk about what a controlled experiment is here in a little bit. We're going to record or analyze the data that we get. We're going to draw some conclusions from that data. What does that data mean? Did we support our hypothesis or did we disprove our hypothesis? And then, depending upon our conclusions, we'll either accept our hypothesis or we'll form a new hypothesis and retest. So step 1, ask an observation or ask a question. So make an observation, ask a question. Okay, so there are some examples for you. Step 2 is form a hypothesis. And remember, this is simply a prediction that has not yet been proven or disproven. Okay? Remember that it has to be testable. Testable means that evidence can be collected that either does or does not support the statement. Step 3, we're going to design a controlled experiment. So when we talk about a controlled experiment, one of the words that we have to talk about are variables. And variables are things or factors that can be changed within an experiment. changing the temperature, changing the amount of light, changing the concentration of the solution used or the fertilizer, changing the amount of time before measuring. Okay. The key that makes this a controlled experiment is that we only alter one variable at a time. Okay? If we were to alter multiple variables at a time, we don't have any way of knowing which one is responsible for the results that we see. Okay? That's really, really, really important. So what that means is that we need to have a control group and an experimental group. One group that doesn't have anything changed, it's kind of like our baseline, and then another group where we're going to change a variable so that we can see the difference in the results. Oh, let me go back. Okay, give you a chance to read that one. Okay, so here's an example. So in order to test the effectiveness, and this is completely not realistic, but in order to test the effectiveness of a new vaccine, 50 volunteers are selected. We divide them into two groups. One group is given the control, and the other is the experimental group. Okay? Both groups are given a pill that is identical in size, shape, color, and texture. Now, the one difference is going to be that one group, there's not going to be any vaccine within the pill. There's going to be no vaccine component to the pill. That will be our control group. The other group will be the one with the vaccine. Okay? So, I just did this for you. Describe the control group. That means the experimental group will have the vaccine within it. Now, what variables are kept constant? What stayed the same? And what was changed? Okay. There are two variables in an experiment. We talk about an independent variable. That is the thing that got changed by the scientist. And there's a dependent variable. That is the thing that we measure. That is the thing that we collect data on. If we're doing a quantitative experiment, then we're going to measure and collect that number. If we're doing a qualitative experiment, then we're going to observe and record that qualitative data. Both are perfectly acceptable. So I want you to think back to our example. So what was the independent variable? The thing that was deliberately changed. And what was the dependent variable? What was the thing we were looking at, that we were measuring, that we were quantitating or qualifying? Okay, so your independent variable was the addition of the vaccine to the pill. That was your independent. Your dependent variable was the observation of the person's health. Were they healthier with the vaccine or not? Then we record the data. And we analyze it. Does it or does it not support the hypothesis? Just remember that data does not have to support the hypothesis to be reliable or valid. We run experiments where the outcome is not as we expect all the time. Data is data. You should not be emotionally connected to the data. And reliable data often doesn't support the hypothesis. Think of how many times Edison tried to make a light bulb. Okay? He had a hypothesis. He thought this would work. He gave it a try. It didn't work. After numerous failures, he said, okay, well, I know all of these ways to not make a light bulb. So his hypotheses up until that point had all been incorrect, but the data was the data, and it was still valid because of that. We draw conclusions. This is very important. We need our experiments to be well documented so that other scientists can repeat them. we don't consider them valid until multiple different individuals in different settings can repeat and get the same data. And once we have a hypothesis that is repeatedly supported by the same experiment and the same data is acquired, we can talk about forming a theory. And a theory is just, it's a really broad, it's a comprehensive statement of what we believe to be true. Okay. There's our blue box about theories. Okay, here's our practice problem. Ready? Okay, so you're going to design an experiment to test different concentrations of fertilizer. You have a weak concentration, a medium concentration, and a strong concentration, and you're going to start with, I believe, 100 orchid plants. So, what's your hypothesis? My hypothesis is that if I use a medium concentration fertilizer, then the orchids will grow better than if I use a low or a high concentration of fertilizer. So what I'm saying is that if my concentration is too low, it's not enough to benefit the plant. If my concentration is too high, then that's too much and that will damage the plant. So what I want is a medium concentration. Your hypothesis could have been different. They don't. I'm just giving you an example. So then if we assume that we're using mine, let's set up a controlled experiment. So here's what I would do. I would take those 100 plants, I would divide them into 4 groups. Why 4 groups? Because I need 100 plants to be divided so that I have 25 that are going to get plain water, no fertilizer. That's going to be my control group. Then I'm going to have 25 that are going to get a low concentration, 25 that will receive a medium concentration, and 25 that will receive a high concentration of fertilizer. Okay? And then I need to keep everything else the same. Everything else needs to be equal. That's how this is going to be controlled. My variable that's being changed, my independent variable, will be the amount, or the concentration of fertilizer. Okay? Everything else needs to be the same. So I have a control group and an experimental group. Who's my control group? Who is my experimental group? So my control group is that group of 25 plants that just received plain water, no fertilizer. My experimental group, I actually have 3 experimental groups. Well, I have 1 experimental group with 3 subgroups. So I have 75 plants that get varying concentration of fertilizer. All right, remember what I said. We have to keep all the variables except for that independent one, so we have to keep everybody else equal. The other one that I didn't type on there that I probably should have is that they all have the same the same soil and the same amount of soil. Okay, so here's the data after 1 month. So is that hypothesis supported or disproved? I said that the medium concentration fertilizer would produce the most growth. And no, it wasn't. That isn't what happened. It actually appears that the weak concentration of fertilizer. So my results do not support my hypothesis, and my hypothesis is disproven. I'm going to give you that same information, and I'm going to ask you a different question now. What is your conclusion? Remember, we have to conclude and analyze and conclude. Okay, so let's do a little bit of review. didn't exactly talk about this, but you want to have a large sample size. You wouldn't want to test this with just 4 plants or even just 8 plants. We chose a large sample size of 100 plants. If it's too small, we get an inaccurate conclusion. We can get false data. So you want a large, it's called an N number, but you want a large sample size. So we repeat experiments to see if we can produce the same results, to make sure that it's not a fluke. Every time that we reproduce the same results or very, very similar, statistically similar results, we consider that to be a supporting body of evidence, and that leads to the validity of our results. What's the importance of a control? Well, that control helps us to tell us when the experiment, what will happen if the experimental factor was omitted, i.e. no fertilizer at all. So without the control, I have no basis for comparison. I don't know that the fertilizer makes a difference or not. How is a theory different from a hypothesis? This is very important to understand. So hypothesis is just a guess. It's an educated guess, but it's a guess. It's one explanation or possible explanation for an observation. Remember that it has to be testable. A theory is a broad statement that evolves or comes into being after we've had several rounds of experimentation and we have considerable data collected, and all that data is supportive of the hypothesis. Why do we need good record keeping? Why do we need scientists to accurately describe their procedure? Remember what we said, it needs to be able to be repeated by others so that it can be verified and considered valid. Know your difference between independent and dependent variables. When you graph these, the independent variable is going to, I gotta think this for a second. The independent variable is going to be on the x-axis, and the dependent variable is going to be on the y-axis. Just remember those definitions. Remember why we have to keep one variable, only one changes and everybody else stays the same. Okay, that's the end of our slideshow. In some sections you have a quiz on this, in other courses you do not. Please feel free to review this at any point in time. If you have any questions, let me know. Thanks.